Regulation of NLRP3. ATP is the energy source of our cells, and like many proteins, NLRP3 is powered on by ATP. It makes sense then that people with CAPS have higher than average ATP binding to NLRP3, but there's more to the story when it comes to regulation of NLRP3, and that's what I want to delve into a little bit today. So brace yourself and let's do some science. This shape represents the NLRP3 protein, and above the protein I've lined up the exons from the messenger RNA. So if you have a mutation in exon 3, you can see it is likely to be in the NUC domain of NLRP3, which is where most CAPS mutations are. But if your mutation is in, say, exon 1, then your change is in the PYRIN domain. I've got my mutation shown here in the NUC domain, my alanine to valine change at amino acid number 439 in the string of 1,036 amino acids that make up the full NLRP3 protein. It's critically important that NLRP3 is regulated so it's not activating all over the place, and there are many ways this happens. I'm talking about one type of regulation called post-translational modification. Translation is the fancy science word for when our bodies make proteins. So these are modifications after translation, which just means they're some of the things that can happen to the actual NLRP3 protein. Have a look at the red and green P's, the purple UBs and the pink S. P stands for phosphorylation. It's a phosphorus connected to some oxygens that, with the help of an enzyme called a kinase, drops an oxygen and attaches itself to an oxygen on a protein. Amino acids make up proteins. The only amino acids that can be phosphorylated are serine, THREONINE and TYROSINE because they're the only ones with oxygens. S is the code for the amino acid serine, and you can see a phosphorylation site at amino number 3 in the PYRIN domain of the NLRP3 protein. I've coloured it red because it is a deactivating modification. It stops the PYRIN domain binding with the PYRIN domain of another protein and prevents formation of an inflammasome. There's a green phosphorylation site on a serine at position 196, which is an activating modification because it influences removal of the UBs, which I will explain shortly. There are both red and green Ps at position 293 because phosphorylation at this site can be involved in activating or deactivating NLRP3 depending on which kinase enzyme attaches them. Then we have a red deactivating phosphorylation site down in the leucine-rich repeat domain at position 859 on a tyrosine, shown by its single letter code Y. So we have some on and off switches. And remember, this protein comes out and folds up. It doesn't sit in your body in a straight line like my picture. When you have a phosphate group sitting in a protein, it can change the shape of the folded protein so that it's blocked or activated, or varying levels in between those on and off states. Don't think of it as an all or nothing situation. Regulation is about maintaining a balance, and if you want the fancy word for that, it's homeostasis. Now look at the purple UBs. We have one attached to a lysine, amino code K at position 46 in the PYRIN domain, and there's one attached to a tryptophan, amino code W at position 71. UB means ubiquitination or ubiquitylation, depending on who taught you science. What they are best known for is tagging proteins for the recycle bin. That's right, you've got recycle bins in your cells, technically called proteasomes. When these UBs are put onto NLRP3, it gets sent to the proteasome recycle bin and pulled apart. The various bits get reused elsewhere. The pink S down at the lysine shown by its code K in position 689 is a SUMO. SUMO stands for small ubiquitin-like modifier, so you can think of it as a different type of UB, and this one is also inhibitory. So, now you're an expert in NLRP3 post-translational modifications. I'm kidding. This is an active area of research, and by saying that, I mean our understanding can change as new discoveries are made. But think about how many different mutations can cause CAPS. Most of them might be in exon 3 and involve enhanced ATP binding, but NLRP3 mutations can and do happen all the way down the protein. What are the chances that some of them affect some of these post-translational modification sites? A new drug that targets the ATP binding site of NLRP3 might be great for some of the mutations, but might not work for others. This is why it's important to understand how the different mutations are causing disease in the first place.